Hello, my name is Joshua Mugabi. I welcome you to the origins of cells. Lesson objectives. We are going to look at non-living synthesis, Miller-Urey experiment, biogenesis, pasture experiment, RNA world hypothesis, endosymbiosis, and oxygenation of Earth. Welcome. Let's start with the functions of cells. The primary function of a cell is to maintain an internal environment that is optimized for metabolic reaction. So we are looking at the primary function of a cell. And we are saying the primary function of the cell is to maintain an internal environment that is optimized for metabolic reaction. So what we are saying here, that the cell is responsible for the keeping of the internal environment. It has to keep the internal environment so that it is conducive for metabolic reactions. Metabolic reactions or metabolism is a sum of all the cellular reactions in a cell. It is the sum of all the cellular reactions that occur within the cell. So what happens is that the cell, our cell, is going to be provided with nutrients. We shall get nutrients that are coming from outside. These ones are provided to the cell. Then the cell carries out what we call metabolism. And this metabolism is the one that results in the production of energy that is going to be used. And the undigested materials are removed out of the cell, which we are referring to as the waste. So, for that reason, we say the function of the cell, the primary function of the cell, is to maintain an internal environment that is optimized for metabolic reactions. This metabolism or the metabolic reactions are necessary to sustain life functions. So all the metabolic reactions that are taking place in the cell, they are important for the maintenance or the sustenance of life functions. And this one is done via the production of biological energy. And that biological energy is what we are referring to as ATP. So all the reactions that are taking place in the cell, all those reactions that we have in the cell, they are for sustenance of the life functions, which we talked about, Mr. Shen. Metabolism, reproduction, sensitivity, homeostasis, excretion, nutrition, and growth. All those are life processes or life functions. Those life functions to be sustained, we need metabolism. And this metabolism is going to result in the production of energy. And that energy is the one that we use to to sustain the life functions. This includes the capacity for self-replication. It includes the capacity for self-replication. So the cell has to make other copies of itself. And that one, of course, is going to require also energy. Cell formation, non-living. It is believed living cells arose from non-living. There is a belief that living cells came from non-living materials. How? They say there was non-living synthesis of organic monomers. There was a non-living synthesis of organic monomers. So which means that one time they were inorganic compounds, and then these inorganic compounds produced the organic compound. It's what they are saying. It is believed the cell rose from non-living material. Non-living materials, we have the inorganic compounds. And these non-living synthesis of organic monomers. The organic monomers were produced from non-inorganic compounds. That's how it is believed. And then the organic compounds assembled themselves, and then they formed complex polymers. So they are saying that these organic compounds that were formed, or the organic monomers that came from inorganic compounds, they assembled themselves and formed the complex polymers. After assembling themselves, they formed what we call the complex polymers. That is how it is believed. Then, later on they say that certain polymers, they became capable of self-replication. They could make themselves. So, they say that first of all, these monomers, they turned into the polymers. And now these polymers were able to replicate themselves to form very many of themselves. That's now we are going to cell formation. But those cell formation, they are saying they are coming from inorganic compounds. Inorganic compounds which later on formed organic compounds. Then the organic compounds became the monomers and formed polymers. And these polymers were able to carry out self-replication. These molecules became packed into membranes. So they are saying that after these polymers forming, they packed themselves into membranes. They started packing themselves into membranes. And this precursor, protobiotics, gave rise to simple cells. So after the molecules forming from polymers, they formed the cell membrane. They formed membranes. They were packaged in a membrane. And now, these ones were the precursor for the rise of the simple cell. So that's how it is believed that the cells were formed. Starting from the inorganic material, forming the organic monomers. Then organic monomers formed the polymers, which polymers were able to replicate themselves. And then when they had finished replicating themselves, they became packed in membranes, and after packing themselves into membrane, now these ones, they rose into what we call the simple cells. Now, when we come to the Muller-Urey experiment, remember we are talking about the origin of cells. Miller and Urey demonstrated the non-living synthesis of organic materials by creating prebiotic earth condition. So here, Muller and Urey, what they were trying to prove was that they can create organic materials by providing conditions which were thought of that there wasn't any life-supporting conditions or prebiotic earth conditions, how the cells would have come up. Muller and Urey, first what they did, they boiled water to vapor, and this was done at a high temperature. So they had an experiment here where they placed water there, and that water was heated at a very high temperature. Now, after the water was heated at a high temperature, here, vapor was given off. The gas, this vapor which was given off, rising, it was mixed with a gas. That's what they say. That the vapor was mixed with gases in a reducing atmosphere. That means that this gas which they added to the vapor did not have any life-supporting gas in it. So the vapor came from down and it got mixed with the gas which did not have oxygen. Now, this mixture of the gases and the vapor, the mixture was exposed to electrical discharge. So when it came from vapor here, which was coming from boiled water, moving upward, and then it was mixed with the gases, it moved up and went into the chamber where it was exposed to an electrical discharge. Here, the vapor mixed with the gas was exposed to an electrical discharge. Now, that mixture in that chamber of an electrical discharge was finally cooled and was left to condense. So from this area here, our vapor was allowed to be cooled. It was taken to this area and it was cooled, and then after cooling it became condensed. It turned back into the liquid form, and when it turned into the liquid form, now it came and collected down here. So what did they find out? They found out that the condensed water here, that water which was condensed there, contained organic molecules. The water contained organic molecules. It had organic molecules. Now, remember, we started with water from this area. Just water. Water is inorganic. And this water was heated at high temperatures. As it rose, it was mixed with gases which were in a reducing condition, meaning that there was no oxygen. The gases mixed with the vapor, they were now exposed to sparks, electrical sparks here. And when they were exposed to electrical sparks, later on, they were taken in a condenser where they were cooled. And when they were cooled, now they came down and collected them down in this chamber here. Now, where the water was collected down, they found out that this condensed mixture, the condensed mixture, it contained organic molecules. Yet in the beginning, we started with inorganic molecules. That was Miller and Urey's experiment, trying to show that it is possible to create organic materials from non-living conditions. Organic synthesis. Now, the initial synthesis of organic compound required key environmental conditions. It required key environmental conditions. One, there had to be a reducing environment. And when we say a reducing environment, that is an environment which has no oxygen. There is no oxygen. But there has to be reducing atmosphere, no oxygen, and there has to be an energy source. There had to be an energy source. And that one, in Muller's experiment, he provided it using heat energy and electrical discharge. So those were the very key environmental conditions that were required in the initial synthesis of an organic compound. We needed a reducing atmosphere, that is an atmosphere without oxygen, and then there was need for a source of energy. Now, at the beginning, where there was no experiment, where were these non-reducing conditions coming from? This non-reducing condition or non-reducing atmosphere, where there was no oxygen and energy sources, it is believed that this condition might have been present in a number of locations on the Earth. And those locations, they think they might have been the hydrothermal vents and the volcanoes. So these vents, the hydrothermal vents, these ones are believed not to have any oxygen in them. And in the volcano, these volcanoes, here we have a lot of supply of heat energy. So that's what they are saying, proposing that the organic synthesis, these conditions which are required, the reducing atmosphere, might have been present in the vents. In these vents, we don't expect to find their oxygen. And also in the volcano, the volcanoes might have provided the heat energy or heat source that was provided for the synthesis of organic substances. Biogenesis. Biogenesis. The conditions required for non-living synthesis of cells do not commonly exist on modern Earth. Now here, people coming in, other proposals coming in, saying that the conditions required for non-living synthesis of cells do not commonly exist on modern Earth. Conditions where we expect no oxygen, no air, they don't exist on the modern Earth. So that we synthesize cells using non-living synthesis methods. They continue to say that living cells cannot arise independently by abiogenesis. They cannot rise independently by abiogenesis. They say that living cell cannot just come from nowhere by abiogenesis where there are no conditions. Cells can only be formed by division of pre-existing cells. Cells can only be formed by division of the pre-existing cells, meaning that a cell is going to come from another cell. You can't make a cell by using conditions, the other conditions they were saying. So they are saying that the cells can only be formed by division of the pre-existing cells. And this law of biogenesis is largely contributed by the work of the French microbiologist, that is Louis Pasteur, who lived between 1822 and 1895. For him, he says, cells cannot be formed from other things that they were saying, but cells can only be formed by division of cells that are already existing. That is Louis Pasteur's work. In his experiment, Pasteur's experiment, he used the method. Broth was boiled, and when it was boiled, it killed the existing microorganism, and that one was referred to as sterilized environment. So for him, he got the broth, boiled it, and by boiling it, it means it was killing off the microorganism. And that one was sterilizing the environment. Then, in a control experiment, the broth was cooled. Okay? The broth was cooled. And when it was cooled, the water collected in the delivery tube. And this water, which was collected here in the delivery tube, it sealed off the mouth of the flask. By sealing off the mouth of the flask, it means no any microorganism would pass here to go into the broth. And when that one was done like that, therefore the conditions inside the flask were sterile. And what he found out that there wasn't any growth of any organism in the broth. When there was no any conditions of oxygen, air inside here because of the blockage of the delivery tube by water here. So microorganisms would not enter into the broth. And because they could not enter into the broth was sterilized and there was no any growth of the material or any organism there. Now, in his experiment, when he got the neck of his experiment. When that neck was broken, it allowed air to enter into the broth. Now, that air which entered into the broth was carrying microorganisms, and those microorganisms entered the broth. When they entered the broth, there was contamination. The broth became contaminated. And after a short time, he found there was growth of microorganisms in the broth. Yet remember, in the other experiments he carried out, whereby there wasn't any breakage here, no breakage in the apparatus, and he had sealed off here this entry with the water. There wasn't any entry, there wasn't any contamination in the broth, and no organism was found there. But here in this experiment, where he did not break off, where he broke off the apparatus here and did not put any covered entry there, he found out that there was contamination, and then there was also growth of microorganisms. Pasteur's experiment therefore showed that spontaneous generation of cell does not occur on Earth. That was the experiment of Pasteur. It proved that, or it showed that, there can't be spontaneous generation of cells on the Earth, whereby cells are just coming from no anywhere and they appear. It is impossible. That is what Pasteur's experiment showed, because of the experiments he carried out. Heating the broth, sterilizing it, covering the entry with water to prevent any entry of microorganism, and he found out there wasn't any growth of microorganism in the broth, because the environment did not have any living condition. But when he got that same experiment and he cut off part of the experiment and allowed the contamination microorganism to enter into the broth, they found out that there was growth of microorganism in the broth, because there was contamination. And that one showed that organisms or cells can only come from existing cells. They can increase in number, multiply because of the existing cells. And that one was proved by this experiment. And this experiment, the control experiment, proved that the cells cannot just come spontaneous. If they would come spontaneously, in this control experiment, we would have expected to have found their growth, but there wasn't growth of any microorganisms. Chicken and egg puzzle. Chicken and egg puzzle. Now, I want us, before we go through the chicken and egg puzzle, there is this puzzle we used to discuss that an egg and a chicken, which came first. Now, we have seen Louis Pasteur saying that cells come from pre-existing cells. Now, this one brings us to the chicken puzzle. We are saying that the replication of cells require chemical instruction that must be stored as code. Replication of cells This is the making of new cells by the existing cells. But the cells that are existing to make new cells, they need what we call a chemical instruction. They need what we call a chemical instruction. And that instruction is always stored on what we call a code. So the chemical instruction is needed in order to give instruction to the cell to replicate or to make other cells. Now, the DNA is the one that functions as the genetic code. It is the one which has the chemical instruction which is required by the cells to produce other cells. And why at the same time we have the proteins are the ones that are doing the chemical work. So I want us to understand here. We have DNA. DNA is the one which is the genetic code. And this genetic code is the one which has got the chemical instructions. These chemical instructions are the ones that are going to help the cells to produce other cells. Now, where is the problem arising? Remember, we are looking at the chicken egg puzzle. DNA is having the genetic code, which is going to end up helping in the production of the protein. The proteins are the ones that are doing the chemical work. Proteins are the ones that are doing the chemical work. Now, this chemical work is the one which is going to result into the new cells. The new cells are going to contain the DNA. So now the question is, the puzzle we are having, the DNA and the protein, which one comes first? Remember, the proteins, they require DNA in order for them to be synthesized. But again, that DNA requires the proteins in order for self-replication. If the DNA is to copy itself to form other cells, it requires protein. But the proteins, for them to be synthesized, they need DNA. So which one came first? That is the puzzle. So, proteins and DNA, which of those molecules came first in the origin of the cell? Which one comes first? Protein is going to help the replication of DNA, and DNA is going to help in the production of proteins. This one, DNA, requires protein to be replicated. So who comes first? That is the puzzle that exists. If we say cells come from pre-existing cells, which is true, but which one came first? Was it the protein or the DNA? World RNA hypothesis. RNA hypothesis. Now here comes another hypothesis. This one, it proposes that RNA was the initial chemical instruction. RNA was the initial chemical instruction. For them, they are coming in. If we are failing to find out who comes first, DNA or protein, then they propose that RNA was the initial chemical instruction if there is failure to find out who is first between DNA and protein. Now, these are the reasons. You say that RNA can self-replicate, making it an effective coding molecule. For it can self-replicate. It can multiply by itself, not like a DNA which depends on protein. Therefore now, it can be an effective coding molecule compared to DNA, because DNA is going to depend on the protein. But RNA can self-replicate without the presence of proteins. Therefore, it is an effective coding molecule compared to DNA. Secondly, they say that RNA can function as a catalyst, making it a viable agent for chemical work. Now, if proteins are doing that chemical work by being catalysts and so on, RNA can also function as a catalyst. So making it a viable agent of a chemical work, the work that can be done by the protein. So they are trying to say that RNA might have been the initial instruction chemical. Now, RNA has been superseded by DNA and protein. It has been superseded by DNA and protein. How? 1. DNA is more chemically stable compared to RNA. DNA is more chemically stable than DNA. Why? Because it is double-stranded. And being double-stranded makes it a better coding molecule than RNA, which is a single-stranded molecule. Secondly, the proteins, they have greater variability. Proteins have got better variability. They have a greater variability. Why? Because proteins can be synthesized from a combination of 20 amino acids. And that one makes it a better catalytic molecule, because we can form very many enzymes compared to RNA. So that's where RNA is superseded by DNA and the proteins. So RNA remains as a transitional. It remains as a transitional form between DNA and RNA. I mean, between DNA and protein. So that is the original role of RNA. Its original role is to be an immediator or a transitional form that exists between DNA and the proteins. Full stop. That is its role. Endosymbiosis. Endosymbiosis. Origin, remember we are talking about the origin of cells. Now we are coming with another one, endosymbiosis. Now, here we are saying that eukaryotic cells, they are believed to have evolved from prokaryotes. via a process called endosymbiosis. Here they are saying the origin of cells. Remember, we have two types of cells. We have the eukaryotic cells. Those are the cells that have got a nucleus and membrane-bound organelles. And then we have prokaryotes. These are the cells that lack a nucleus and they don't have organelles which are membrane-bound. So here they are saying a cell, eukaryotic cell, they are believed to have evolved from prokaryote by a process of endosymbiosis. So, long time, there was an ancestral prokaryote cell that existed. There was a cell that was prokaryote. That was the ancestral cell. Now, that cell started forming what we call the nucleus. It went on forming the nucleus. The nucleus started forming in the cell, slowly. Okay? Then from there, microorganisms, like for example, the aerobic bacterium came in, entered into the ancestral prokaryote cell. And then also the cyanobacteria also entered into the ancestral prokaryotic cell. Then what happened? Now, these ones, they remained inside the ancestral prokaryotic cell and they were not digested. And then they started performing functions inside the cell. And then the ancestral prokaryotic cell now turned into the ancestral eukaryotic cell. So here they are saying that prokaryotic cells, prokaryotes, became engulfed and they remained undigested. Here, they are being engulfed. They are being engulfed, and here they remain there undigested. And this one contributing to a new functionality. So now when they were engulfed by the cell, when they were engulfed here, they remained inside undigested. And now they started performing different functions for the engulfing cell. And now this engulfing cell now formed the present day eukaryotic organelles. So, here they are saying the origin of eukaryotic cells can be explained by endosymbiosis theory. So for us, they are saying, because we say that there are two types of cells, we have the prokaryote and the eukaryotes. Prokaryotes are the ones that lack a nucleus. They don't have a nucleus and they don't have membrane-bound organelles. So when we are looking here, they are saying the origin of the eukaryote might have been the prokaryotes, which don't have a nucleus. But later on with time, those prokaryotes, they developed the nucleus with time. And after developing the nucleus, then other microorganisms started entering the prokaryotic cell, engulfed microorganisms. When it engulfed these microorganisms, now they were not digested. they remained inside there in the cell, and when they remained inside the cell, now they started doing work for the engulfing prokaryotic cell. And now this prokaryotic cell turned into what we now call the present eukaryotic cell. There are evidence of endosymbiosis. One, they say that the mitochondria and the chloroplasts are believed to have evolved by endosymbiosis. Because this one can be proved by numerous features that are present in these organelles that support the theory. So, they believe mitochondria and chloroplasts might have evolved via endosymbiosis. And they have got features which are present in these organelles that support the theory. Now, when you look at these features, they say we look at the components and the evidence. The first component are the membranes. All these mitochondria, they have double membrane. In terms of antibiotics, they are susceptible to certain antibiotics. When it comes to division, they reproduce via fission-like process. DNA, they have their own DNA. They have a naked and a circular DNA. And when it comes to the ribosomes, they also have ribosomes of 70 S ribosomes, which makes them to have been organelles which were outside, some organs which were outside, which were engulfed by another cell, and they remained undigested, and then they continued to do functions for the engulfing cells. Oxygen enrichment. Now, we have been talking about the origin of cells, and we have seen that oxygen is necessary for these processes to take place. Then how did oxygen accumulate in the atmosphere when it was not there? Here, it is believed that photosynthesis is the only significant source of oxygen production on the Earth's surface. All the oxygen that we are getting on the Earth's surface is believed to have come from photosynthesis. The evolution of photosynthetic cells saturated the environment with oxygen. So they are saying, when there came photosynthetic cells, they started saturating the environment with oxygen. And this resulted in change in the environment. Now, what happened when there was change in the environment because of production of oxygen? The oceans used to have a high level of depositions of ions, which was released from underground water vents. And then oxygen started reacting with iron and forming insoluble deposits, which are the banded iron formation. Now, when iron in the ocean was completely consumed. Now oxygen started accumulating into the air. When it accumulated in the air, the atmosphere changed from being anoxic, whereby it was having no oxygen, to roughly having 20% of the oxygen. Then the atmospheric oxygen levels contributed to the development of the ozone layer. And this led to further change of the Earth's atmosphere. The oceans and rock depositions due to photosynthesis. Now, we have been talking about the increase, how oxygen came in by the cells, photosynthetic cells producing oxygen. And they are showing that it first was in the water. It reacted with iron forming insoluble depositions, and when they were finished, now oxygen accumulated into the air. Now, when oxygen is starting to accumulate into the air, it led to the development of the ozone layer. Now, after the development of the ozone layer, now we are coming to aerobic development. That is the development of aerobic organisms. Now we see that oxygen has accumulated in the atmosphere. So what is happening? So here they say an increase in the oxygen due to the photosynthesis led to evolution of aerobes. So here they are showing that oxygen went on increasing in the atmosphere. And as years went on decreasing, like 4 billion years ago, the first cells were formed. 3 billion years ago, photosynthetic cells were formed, and these ones, photosynthesis started taking place. Then aerobes started developing 2 billion years ago. Then 1 billion years ago, then plants started forming. At around 0.5 billion years ago, then animals started developing. So all these ones, they are just showing the events of the cell formation, how the cell started forming from the origin. So they say the formation of the ozone layer constantly reduced or restricted UV radiation levels. And as the levels went on reducing, this means that the ozone layer facilitated the proliferation or diversification of the terrestrial-based organism. Because of the formation of the ozone layer, it prevented ultraviolet light coming onto the Earth, and this led to the increase in organisms that are living on land, terrestrial-based organisms. My friend, thank you very much for participating in the origin of the cell. We have looked at the non-living synthesis. We looked at the Miller-Urey experiment, looked at biogenesis, Pasteur experiment, RNA hypothesis, oxygen, nation of the Earth, and endosymbiosis. Thank you. Please don't forget to subscribe.